If a frog tadpole loses a limb, it can perfectly regrow bone, muscle, and skin. If a mouse suffers a similar injury, it forms a thick layer of scar tissue, and the healing simply stops. For a long time, we thought mammals were simply missing the biological blueprints, as if the instructions for building complex body parts had been deleted from our DNA as we evolved. But we now know those instructions are still there. Mammalian cells still carry the complete manual for regeneration. But for us, those pages have been archived and put into a deep sleep. The factor keeping those instructions asleep is something we encounter every second. It is the high level of oxygen in the air we breathe. Oxygen is what keeps us alive, but it also creates a physical boundary that stops our cells from shifting into a state where they can actually start the repair process. To see this in action, scientists took limb tissue from a tadpole and an embryonic mouse and placed them into separate controlled petri dishes to observe how they healed outside the body. In standard room air, which is about 21% oxygen, the mouse tissue failed to heal its wound, forming a scar just as it would in the wild. But then the scientists tried a different approach. They dropped the oxygen in the mouse's dish to 12%, mimicking the low oxygen water where tadpoles live. These microscopic cross-sections tell the story. In the low oxygen dish shown at the top, the mouse tissue successfully closed its wound and began forming the specific types of cells needed to initiate the regeneration process. This shows that mammalian cells haven't lost their ability to heal. They are simply waiting for the right cues from the air around them to tell them it's time to get to work. To understand why the air matters so much, we have to look at a protein called HIF1A. You can think of it as a sensor that constantly checks the oxygen levels outside the cell. In frogs, this sensor stays active most of the time, but in mammals, exposure to normal, rich air causes the sensor to instantly shut down. However, look at what happens when the oxygen is lowered. These bright yellow specks show the sensors powering back on and flooding the tissue, proving the cells have successfully changed their internal state. As the sensors turn on, the cell's internal structure changes. The stiff skeleton keeping mammalian cells braced and immobile suddenly relaxes, becoming fluid and flexible. It turns out mammalian cells aren't incapable of healing. Their scaffolding is just too stiff and locked down by oxygen to move into a rebuilding phase. That rigid skeleton also traps a specialized builder protein called YAP, keeping it stuck at the cell's edges where it can't do its job. When the skeleton becomes flexible, that builder protein is finally released. It travels straight into the nucleus, the cell's command center. Once inside, the builder can finally access those archived genetic instructions and begin the first steps of organizing the tissue to rebuild what was lost. But there is a catch. As land animals, we need a lot of oxygen to power our large brains and warm bodies. We can't simply stop breathing for weeks to help a wound heal. So, while the biology for regeneration is clearly functional in a petri dish, our own need for oxygen to stay alive has historically acted as a barrier to our recovery. To get around this, scientists developed a specific chemical stabilizer that prevents the cell's oxygen sensor from shutting off. This chemical essentially tricks the mammalian cells, making them behave as if they are in a low-oxygen environment, even when they are sitting in a normal, air-filled room. This chart shows the result of that trick. The first column shows no healing in normal air, but the second column shows how this chemical hack allowed the cells to start the regeneration process in the exact same environment. This is an ancient evolutionary trade-off. Our ancestors gained the high energy needed to live on land, but in return, the rich oxygen we breathed essentially put our regenerative abilities into a permanent hibernation. While we aren't yet at the point of human trials, this research shows that the fundamental secret to regeneration isn't something we have to invent from scratch. It's a matter of engineering a way to speak the right language to the sensors we've already carried within us for millions of years.